Yes, technology for you. It's always a great honor to be up on this stage. Every time I come up, then I'm always wowed for two reasons. One is that usually I get to follow George, and over the last 10 years, and it has been 10 years, then to see the development of the portfolio and see the products that are out there in the marketplace, and to see the work that his team is doing deploying those and the goals they have for the next 5 years is humbling. And the second is, I think Ben mentioned earlier, Ben Rolfe mentioned, it sounds like you're preaching to the choir here. So I'm actually presenting everybody else's data and everybody else's accomplishments, and actually everybody else's goals for the next 5 years. So this is actually this is the key slide, and it fits. I was listening when Marcel was talking about, how does this fit with the Malaria Roadmap, which you saw on, I think, David's slide 8, where you have different countries which are going, reducing from high to low transmission, from low to zero, maintaining zero. It does actually fit over the top of that. This is more driven by operational practicality, what you think could be done in the next 15 years. And what you'll see as I go through it is how some of those apply to different bits of that malaria eradication agenda. So they do fit together, but they're not as intuitively obvious as sometimes we would hope. Okay, so when we talk about the case management for uncomplicated malaria, as I said, there are a number of things that are going on at the moment in terms of expanding, putting more compounds into the ACTs, but the real goal is when can you come out with the next generation of medicines which will simplify therapy and fight resistance. So all the time, just to reassure everybody, the number one rule here is that every medicine that we develop, everything we take into clinical development, of course, must be active against all existing resistant strains. So what we're talking about here is, making sure our new medicines are future-proofed against new resistance coming forward. This is actually where the bulk of our development candidates have been funneled, certainly over the last 5 to 10 years, and you'll start to see there's a little bit of a pivot to saying how can we move some of our candidates that come out into some of these other boxes. Case management of severe malaria, then we've done very well over the last 5 years with partners moving forward artesunate injectables, artesunate suppositories. The question would be, if the resistance got worse, so you saw it from Rob's presentation, that patients can be treated with 6 days of artesunate at the moment, and that will work quite well. If we move to a new level where, say, artesunate resistance sees an IC50 shift, a classical resistance, we're then going to go back from artesunate injections back to quinine. So then the question is, what would you have that would replace? We have a whole pile of molecules, starting with KE609 from Novartis, which are actually very, very fast acting. So that's one of been our screening points. The question is now, which one of those can be formulated as a new injectable? And also, how do you develop as an injectable, given that you can't go forward and do a big 15,000 patient study as was done with artesunate? This is why we're here in Bali today to talk about relapsing malaria. Not just about the relapsing of Vivax. Don't forget, we also have relapsing of Ovali in Africa. So what are we going to do about the dormant forms? And can we please have a drug, everybody says, it's the plea from this part of the world, can we please have a drug that doesn't have a G6PD liability for the future? One of the things to say here, and this comes up a lot, is that we're actually quite fortunate in malaria that we know we can target the dormant reservoirs. So if you go to an HIV meeting, you go to a TB meeting, they don't know how to do it yet. So we know it can be done, we just don't know whether it can be done with a different chemotype than the 8-aminoquinoline. And that's what we'll be talking about when we get to Project of the Year. What we should know within the next 5 to 10 years is actually whether this is going to have any legs. Clearing the human reservoir, so this is the low-dose primaquine. Again, all of our molecules are screened for transmission blocking activity. What we're looking for now, since we have WHO guidance on low-dose primaquine, is saying, how do these molecules in the pipeline compare with low-dose primaquine? In other words, can we get away with not having to give low-dose primaquine in the future just because all of our drugs have that activity? And then the final two at the bottom are really interesting, the way that the thinking has evolved. So chemoprevention, I think there's been a love of a lot of people in the room for a long time. Say the idea that you can give children medicine and that protects them from getting the disease, knowing that we don't have a highly efficacious vaccine. This is a fairly obvious way forward, but you can see from the pathway that it took, this was quite a long journey. So looking now and saying, okay, this is, as George said, we do this in the Sahel. What's the next generation drug? How do we make it more convenient for patients? And how do we look at what's going to come after that? Chemoprotection, that's taking people who are immune normal, so travelers into an area, always used to be a no-no. Right? We're not here to protect the US Army when they go into malaria endemic zones. We're not here to protect tourists, you know, they have their own markets, whatever. Until the malaria eradication agenda said, well, wait a minute, you know, as you start to eradicate malaria, then what you will get is you get more and more populations which are completely non-immune, migrating into areas with high malaria. So you do actually need ways of dealing with those populations, and you'll also need ways of dealing with epidemics when they come up. What's happened in parallel, of course, is that fields like HIV have gone in. HIV has PrEP, so pre-exposure prophylaxis, has said as well, you know, we could do chemoprotection with drugs. We could give an injection that will protect for 2 months, 3 months, 4 months. And actually, if you could get that sort of protection, this would be really valuable in the field. So that's effectively my 10 minutes. As George says, the pointer doesn't... Okay, so just to take you through quickly. So in terms of case management, the two things, and we'll talk about this tomorrow, are can we simplify therapy and can we overcome resistance? At the moment, then, the therapies are working quite well, apart from in Cambodia. So simplifying seems to be a very useful goal there. One of the interesting things there is when you look at the portfolio, there are actually 14 new candidate molecules, and by new, most of those have been discovered in the last 10 years, so since 2007, by quite a lot of the people in the room here. So they also target, they have new targets, they have new mechanisms of actions just because of the way they were found. So presenting this is not just a numbers game in terms of how many molecules we have, it's also the diversity of the portfolio is getting higher year on year. We look, I've talked through most of this, but to basically say we have deliverables over the next 5 years, and they're aspirational goals around those 5 areas. I've talked through those, so I won't repeat now. But just to say, getting new combinations, move forward, moving new classes of molecules in, having something for severe malaria, basically rolling out to tefenoquine, and then starting to look at what the next generation chemoprevention would be after SP amidiquine. In the middle area, so we have a product development group, we have a translational sciences group, we have a discovery group. The translational sciences, I'm looking for James here. I know he's gone. we have benefited a lot from having models of human infection going back into that to be able to streamline our drug development. Not in every case, but at least to be able to streamline. So basically what we're looking at in the next 5 years is building on that experience. So having new molecules to take into blood stage infections, making sure we can actually get the transmission blocking data for new molecules in people. So this is not in test tubes, this is people. We have lots of transmission blocking data. Every compound has it. In vitro, in membrane feeding assays, but just understanding what that means clinically is important. And then saying, looking at new molecules going forward for chemoprotection, that means preferably having liver stage activity, and actually getting that data in sporozoite challenge models. So you can see here then we're overlapping quite strongly with what's going on in the vaccine community. We're using the same models. I'll talk about this tomorrow, but just to say coming forward, actually this is a prelim for tomorrow for some of you if you're not here, but just to say, I showed you 14 molecules. One of the challenges is how you put them together. So 5 years ago, we didn't have too much of a problem because we only had 2 or 3 molecules in translation. So combinations of 2 or 3 molecules when you only have 2 or 3 molecules is mathematically very simple. Now you have 10 or 12, the question is, how do you pick the best ones? And that's one of the things we'll be building up is our technology. So we have a combination selection tool, a way of working on that. We can look at drug interactions in vitro, that's what we do at the moment. And we're building our SCID combination work. We do that piece by piece at the moment, but just to get a whole picture. And one of the things we've noticed is that MMV therefore becomes the trusted broker of all that information between our partners. This is the I've lived in development for 10 years. I came from discovery, so this is the development perspective of what discovery people do. They develop new candidates. Nobody knows where they come from. I'll talk about that tomorrow. But to say, we're actually quite lucky at the moment that we are still getting 2, maybe 3 development candidates per year going into the portfolio. There is no reason to believe that that productivity should continue endlessly. In other words, the return on investment, the amount of work we have to do to get a new candidate, or how creative we have to be, is going to seriously have to go up. But we have 2 or 3 new candidates coming in, so that keeps us at a reasonable steady state. So what do we want out of those new discovery things? So we can dream here, and in discovery then they tend to be much more long-term things. But so, as I said, all the drugs have to address drug resistance, including what's going to happen in the future, something about transmission blocking, and something about making sure that we're simplifying therapy. So can we get to single exposure chemoprotection? As I said, then what we're doing here, this is back in discovery, is continuing to invest in the transmission of the malaria parasite, the P5X hypnozoite, and the liver schizons. So it's worth saying at that point, one of the things that we went to the board, the MMV board, and said was, at the beginning of this business cycle, was saying, well, there will come a time, hopefully, when we don't need any more drugs. There will come a time probably in 2025, if the WHO projections are true, 2025, then malaria will be less of a problem than schistosomiasis is today. So it's going to get harder and harder to discover drugs, and it's going to get harder and harder to fund the discovery of drugs. So one of the things that we said is, for the next 5 years, probably for the next 10 years, therefore we have to push very, very hard to make sure that we resource discovery and we make sure we've got the molecules coming through. And I think MMV is very different from the other PDPs if you look at the balance, the sheer amount of resource we put into discovery. So the priorities are to get the molecules coming forward, so 2 from preclinical development. We spend quite a lot of time advising these days on projects that we're not actually funding. And of those 2 or 3 molecules, preferably having one that has transmission blocking activity and one that has anti-relapse activity over the next 5 years, and looking at ones that have liver stage activity and long-acting formulations, so they could be used as injectables. So that's the malaria equivalent of PrEP if you're an HIV person. On top of that, there's a whole pile of platforms, and I don't want to go through all of these, but just to say, it's basically saying continuing to invest in P5X, knowing that without the elimination of P5X and also Bali, then we're not going to get the eradication of malaria. Secondly, target-based discovery. We had a big push, largely led by Elizabeth in the room, to actually do high-throughput screening based on phenotypics. That's led us back into new molecular targets, again, a consortium that she's leading. But to say we have new molecular targets, there are new ways of looking at things, but also we have new chemotypes where we haven't managed to raise resistance. So I call them irresistible. The chemists get really upset about that. But just to say, we're looking now, can we develop new classes of compounds where it's hard to develop resistance? Backup strategy, I didn't talk about attrition. You know, part of the problem of our lives is that in drug development, then things fail. So 9 out of 10 things that make it into phase 1 do not make it out the other side. But every time they fail, you learn something. And so the question is, when something fails, you go back into the catalog of compounds you have, you see, do you have a compound that has that activity without that weakness? So actually moving forward, because we have 10, 12 compounds in development, then looking at the backup strategy and making sure that when we have an issue, we check back, do we have a compound without that issue, is going to be very important. Next generation technical platforms, say intramuscular formulations I've talked about, endecticides we can talk about over coffee, but basically say this is the ivermectin, can we get something that actually blocks transmission by preventing the insects from surviving? And then, as Alan talked about, you know, how do we make sure that our data sharing means that everybody gets access to all the information? So everybody believes that in the days of the internet, then everything is easily accessible to everybody, and it's simply not. So one of the questions is, how can we encourage not only our data to get out there, but the rest of the community? So I shall finish there. And as it started, then just to thank everybody in the audience, because it's actually your presentation, it's your roadmap, it's working together with you that's enabled us to achieve this pipeline, and working together with you which will enable us to take it forward over the next few years. So thank you.